Good day, Bid 4A. Hello, let's continue our discussion for today. For my part, I will be discuss and explain to you the things that I know in my part, which is the continuation of Unit 9, Part 3, the target inhibition of gene expression. I am Mariel YY, your reporter for today. But before we start, I prepared a short icebreaker. So let's play a game. Welcome to the games. The title of the game is Zoom It In Gene Therapy Edition. So, how to play the games?
How to play a game. First, look at the picture carefully. Second, write down the name of the picture. Name should be in the correct spelling. Third, there are 2 rounds, easy and difficult. And last, the person with the most points will win the game. Are you ready to game a play? In picture 1, guess what's in the picture? Are you done? So, let's see if your answer is correct. The answer is gene. How about the picture number 2? Guess what's in the picture? Let's see if your answer is correct. The answer is disease. Next is the difficult round. Guess what's in the picture number 3? Let's see if your answer is correct. The answer is RNA. How about the last picture? Guess what's in the picture? Let's see if your answer is correct. The answer is DNA. Congratulations to those who got the perfect score. Thank you for playing.
Now, let's proceed to our discussion, target inhibition of gene expression, and we will be focusing and discuss the 3 main subtopics, which is 1. Target gene mutation connection, 2. Gene therapy approaches, 3. Method of gene therapy. And what are those methods and approaches that use in target gene inhibition?
Target inhibition of gene expression. This is to block the expression of any disease gene or a new gene expressing a protein which is harmful for the cell.
In gene therapy inhibition, it is also a therapeutic approach that aims to reduce the expression of specific genes, particularly in treating diseases caused by faulty gene, wherein faulty gene is a gene that has undergone mutation, resulting in an altered or non-functional gene product. The process you will see is visually represent of how a therapeutic gene product can effectively block the expression of a faulty gene, thereby restoring normal cellular function. Wherein dito sa figure na yan, makikita nyo kung paano ba yung step-by-step process ng gene therapy inhibition. Dito sa step-by-step process ng gene inhibition therapy, kung saan makita nyo sa unang process is yung cell containing faulty gene. Itong cell na nagko-contain ng faulty gene ay nag-lead siya ng production kung saan yung protein ay naga-harmful. Then sa cell containing faulty gene na yun, nagkakaroon ng blocking ng gene kung saan yung blocking ng gene na yun ay nag-i-introduce ito sa my cell. Then after ng ma-block na yung gene ng gene therapy is nagkakaroon na naman ng new gene product blocks by faulty gene kung saan dito nagkakaroon na nga ng new gene product interacts with and blocks the faulty gene, preserving it from expressing the harmful protein. Kung saan pag nagkaroon na ng new gene product or block faulty gene, nagpe-prevent na dito yung pagkakaroon ng harmful sa protein. Then after that, yung cell functioning normally, dito naman sa cell function normally, nag-result naman ito ng function na normal, kung saan yung function ng cell ay normal.
To sum up the process or the step-by-step procedure of gene inhibition therapy, it is essential for us to know for the safe, effective, and reproducible of gene inhibition therapy. And also, i-ensure din yung control, precision, at nagkakaroon ng better understanding sa process ng gene inhibition therapy. Mechanism of target inhibition. The primary mechanism of target inhibition of gene expression can be broadly categorized into two main approaches. The first approaches in target inhibition of gene expression is transcriptional inhibition. In transcriptional inhibition, this mechanism targets the process of transcription where DNA is copied into RNA.
Ang transcriptional inhibition ay isang proseso kung saan pinipigilan ang pagpabuo ng DNA sa RNA. Dito rin sa transcriptional inhibition ay pinipigilan yung gene sa DNA na maging RNA. And once na maging RNA, yung DNA ay umuulit lang yung protein na meron yung RNA sa katawan natin. So, dapat yung gene sa DNA hindi pwedeng maging RNA kasi bababa yung chance na mag-growth or yung development ng body ng tao. Then if the DNA becomes RNA, nakaunti din yung co-produce na protein. And if that happen, magkakaroon ng kakulangan sa protein na dahilan na pagbaga ng growth or development at magkakaroon ng X-genetic disorder. Like sa physical aspect ng tao, nagkakaroon ng hindi magandang development. Like mayroong dwarfism person and gigantism person na nangyayari lang yan pagka yung DNA is naging RNA.
Makikita nyo sa picture na kung saan yung may double helix, yun yung DNA, and yung single helix is yung RNA. Here in target gene mutation correction, it is used to correct a defective gene to restore its function, which can be done at genetic level by homologous recombination or at mRNA level by using therapeutic ribozymes or therapeutic RNA editing, which is dinedescribe dito yung process ng gene therapy or the revolutionary approach to treating genetic diseases by correcting defective genes. Also, na-highlights dito yung two main levels which is this correction can occur. Una, yung genetic level o yung homologous recombination, kung saan yung homologous recombination is a natural process where similar DNA sequences are exchanged between two DNA molecules. In gene therapy, this process can be harnessed to replace a defective gene with a functional copy. Then also yung number 2, yung therapeutic ribozymes are specifically designed or RNA molecules that act like molecular scissors, cutting and removing the defective mRNA before it can be translated into a faulty protein, which this prevents the production of defective protein, effectively correcting the gene's function at the mRNA level. In the summary of this gene mutation correction, gene therapy aims to restore the function of a defective gene by either replacing it with a normal copy at the genetic level or homologous recombination or by correcting the mRNA. Also, this gene therapy mutation correction offers promising avenues for treating a wide range of genetic diseases and hold the potential to revolutionize healthcare. Here in figure 5, this is used for disease caused by mutation. The corrected gene will be swapped by the mutant gene X. Then diseased cells will become normally or normal after the correction of mutation by gene therapy. Here is the process of target gene mutation correction. This process depicts of a target gene mutation correction, a type of gene therapy. In here picture, gene X is the figure begins by showing a diseased cell containing a mutant gene X or represented by M. This mutant gene is causing a gene. As can you see, yung gene X, dyan nagmumula yung disease cells or mutant gene ng X. Then, in gene therapy, a normal function
functional copy of gene is introduced siya sa gene therapy. The normal gene interacts with the disease cell's mutant gene X. The diagram illustrates a process of resembling homologous recombination. The normal gene replaces the defective M or gene with the chromosomes. Then, after the replacement, the cell now contains the corrected genes. The formerly diseased cells now exhibit a normal phenotype because of the functional gene is restored. So, yung normal phenotype ng gene mutation corrected to restore a functional gene therapy. So, in short, this process visually represents the gene therapy correcting a genetic defect by replacing a mutated gene with a healthy copy via mechanism similar to the homologous recombination. So the outcome will be the restoration of normal cellular function, which is yung normal phenotype genetic mutation ay nare-restore na siya into normal process.
Now, let us proceed to the methods of gene therapy. The goal of gene therapy research is to determine whether a new gene can be used to replace or inactivate a mutated gene to treat a disease or help the body fight a disease. So, the goal of this gene therapy is that this gene therapy aims to use a healthy gene to either replace a faulty gene causing a disease or to disable or inactivate the faulty gene, which is thereby treating or improving the body's ability to combat the diseases. For a gene to be delivered into a cell, a transporter is typically used. A transporter is known as a vector. So, the transporter is also known as the vector, which is the vector is introduced the new gene into a cell or a delivery system is necessary, which is this delivery system is called a vector. Yun nga, tinatawag na vector yung delivery system. So, a vector are essential transporters that carry the therapeutic gene into the target cells. Different types of vectors exist, each with its own advantages and disadvantages. A vector is made from an altered virus. Before the virus is used as a vector, its viral genes are removed. So, the gene therapy often utilizes viruses as a vectors because of their natural ability to efficiently deliver genetic material into cells. However, the virus is modified to remove its disease-causing genes. This also ensures that the vector is safe and won't cause illness. Once the virus is modified, it is intended to transport the desired gene to a cell without causing disease. Once inside, the desired gene should restore the function of the protein. The function of the protein, which modified virus, now a harmless vector carries the therapeutic gene into the target cell. Once inside the cell, the therapeutic gene is expressed, producing the functional protein that was previously missing or defective due to the target or the gene disorder. This restoration of protein function is the intended therapeutic effect. Also, one of the methods of gene therapy, vectors can be given intravenously, which means they are administered into a vein or injected into a specific tissue in the body. Also, other procedures such as surgery can also be used to deliver vectors into specific areas of the body. Now, we are here in the last part of my report. Two approaches for the transfer of gene in therapy. But for my topic, I will discuss to you the first one of approaches is the transfer of gene directly to cells inside the body, and the other one will be explained to you my group mates. Now, let's know about transfer of genes directly to cells inside the body or in vivo. What is vivo gene therapy? In vivo gene therapy refers to the gene therapy administered directly into the body of a living organism. The therapeutic gene is delivered to the target cells within the patient's body, as opposed to ex vivo gene therapy, where cells are removed, modified outside the body, and then reintroduced. In this mode of gene therapy, genes are transferred to the cells grown in culture. Transformed cells are selected, multiplied, and then introduced into the patient. This process involves transferring the desired gene into the cell grown in the culture. So these cells are then selected based on their successful transformation and are multiplied in the lab. Finally, the modified cells are introduced back into the patient where they can produce the therapeutic proteins or perform the desired function. This approach is particularly useful for treating diseases that affect certain cell types, such as blood disorders or genetic diseases. The use of autologous cells avoid immune system rejection of the introduced cells. Here in vivo gene therapy, using autologous cells, which are cells taken from the same individual, it eliminates the risk of the immune system rejecting the introduced cells. This is because the body recognizes its own cell and does not mount an immune response against them. This approach is particularly beneficial in gene therapy, where cells are modified and reintroduced to treat diseases. By using the patient's own cells, the risk of rejection is significantly reduced, making the therapy safer and more effective. This approach can be applied to the tissues like hematopoietic cells and skin cells, which can be removed from the body, genetically corrected outside the body, and reintroduced into the patient's body, where they become engrafted and survive for a long period of time. In this part of in vivo inside the body, this approach, known as autologous cell therapy, involves taking cells directly from the patient who needs treatment. These cells are then grown in a controlled environment outside the body, known as cell culture, after being modified or enhanced as needed. These same cells are reintroduced back into the same patient. These methods ensure that the introduced cells are compatible with the patient's body and reduce the risk of immune rejection, making it a safer and more effective treatment option for the patient who have diseases. The cells are sourced initially from the patient to be treated and grown in culture before being reintroduced into the same individual. So this method focuses on using cells that can be easily removed from the body, modified outside the body, and then reintroduced back into the patient. Example na lang yung include hematopoietic cells or blood-forming cells and skin cells. These cells can be genetically corrected in the lab, ensuring the changes are made precisely. Once reintroduced, these cells can successfully integrate and thrive within the patient's body. Potentially, nagpo-provide siya ng long-lasting therapeutic benefits, this approach is particularly useful for treating diseases and affecting these specifically cell types. Transfer of genes directly to cells inside the body or in vivo. This diagram illustrates an in vivo gene therapy process. And here the step-by-step process. First, harvesting defective cells. Cells containing a defective gene are harvested from the patient. Cell growth in culture. These cells are grown in a culture outside the body. Viral infection. The cells are infected with retrovirus carrying a recombinant RNA molecule containing a normal functional copy of the defective gene. The retrovirus is a vector designed to deliver the corrected gene without causing diseases. Then sa reverse transcription and integration naman, yung inside the target cell, the retroviral RNA is reverse transcribed into DNA and integrated into the cell's genome, replacing the defective gene with a functional one. Then sa corrected gene expression naman, the corrected gene is now transcribed and translated, producing the correct protein. Cell selection and expansion, the cells with a successful integration or integrated and expressed corrected gene are selected, grown, and expanded to a large number up to 100 billion cells. Reinfusion, the engineered cells are reintroduced into the patient where they are expected to function normally and correct the genetic defect. The diagram shows this refusion into original patient. So in the essence of this process in vivo gene therapy, transfer the cells inside the body. This method involves removing cells correcting the gene defect in a control and then returning the corrected cells to the patient to provide long-term therapeutic benefit. The use of retroviruses as vector is a common method in gene therapy, but other vectors may also be used. This image shows two methods of gene therapy, or transfer of gene directly to cells inside the body in vivo. Sa method A, directly to the body, which is yung in vivo, this method involves directly introducing a therapeutic gene into the patient's body. A vector or a delivery system, often a virus carrying the corrected gene, is injected or administered intravenously. So the vector here targets specific cells within the body to deliver the therapeutic gene. Then sa method B naman, yung vivo X gene, ito naman yung biopsy cell, or this method involves removing cells from the patient's body, modifying them outside the body, or yung ex vivo nga. Then introducing the modified cell back into the patient. So, in ex vivo genes, our steps are first yung biopsy or removal of affected cells from the body. Ito yung nare-remove yung cell ng patient through a biopsy. Yung vector administration, ito naman yung the cell are exposed to a vector, viral or non-viral, containing the therapeutic gene. So this allows to the corrected gene to be integrated into cells' DNA. So yung transplantation of modified cells back into the body naman, dito naman na modify yung cell are transplanted back into the patient. This is called an autologous transplantation because the cells originate from the patient themselves. Then, yung diagram na to ay ina-highlights lang yung differences between these two approaches, yung in vivo and yung ex vivo gene therapy. So, in vivo gene therapy is simpler but targeting might be less precise. Sa ex vivo gene therapy naman is more complex, requiring cell culture and transplantation, but na-offers yung more control over gene delivery and modification. Pero yung parehong method naman ay ini-aim nila na ma-correct yung genetic defects or introduce yung new genes to treat the diseases. To sum up my report, which is target inhibition of gene expression, is crucial because it provides a powerful approach to treating diseases caused by overactive or aberrant genes. By specifically silencing or reducing the activity to harmful genes, we can potentially cure or alleviate a wide range of conditions, from genetic disorders to cancers. Understanding the mechanism and techniques involved in this field is essential for developing therapeutic strategies and improving human health. And that's all my report for today. Thank you for watching and listening. I hope you learned something from me.